Morning, ma'am.
Good morning, ma'am.
Good morning, ma'am.
Good morning, ma'am.
So let us continue with biological oxidation. Like I was telling you yesterday, that the biological oxidation takes place as the loss of electrons or by the loss of protons. Okay, so removal of proton can happen from molecules like NADH and FADH2, and therefore there can be the biological oxidation. So simply I can say it is an electron acceptor and electron donor redox reaction that is going to happen, generating the conjugate redox base, redox pair. However, this happens at standard conditions of pH 7, 25 degrees Celsius, 1 atmospheric pressure, and the unit molar concentration. So before I move on to the electron transport chain, we shall see the comparison of the energy that happens in the lab and when it happens in the biological system. That is, comparison of the energy released by combustion and by biological oxidation. So we all know what is combustion. Combustion is the complete oxidation of a compound in presence of oxygen to carbon dioxide and water. Now here I am considering the combustion of glucose. Now one molecule of glucose on combustion in presence of oxygen will give into, of course, carbon dioxide and water, will release 636 kilocalories. This is an exothermic reaction. When you have done analysis of glucose, you have seen as soon as you heat, it just gets oxidized. Okay, so that is an exothermic reaction. Lot of heat is generated. We can do this experimentally, determine the amount of energy released, and it is 636 kilocalories of energy. But this amount of energy cannot be captured in the biological systems because it is isothermal. There is no possibility of energy being released and the temperature getting increased. So this energy in biological system has to be conserved in the form of ATP. If, for example, the same reaction happens, that is C6H12O6 plus oxygen gives carbon dioxide and water in presence of oxygen, and only one ATP will be released because it will be one step reaction. And if it is converted into ATP instead of heat, then the amount of energy that we are getting will be very, very small. Therefore, what happens is, in biological system, the same combustion will happen in series of steps, so that in each and every step, energy can be conserved. Now, when I gave the example of the endothermic and exothermic reactions, I told you that the free energy that will be stored in one molecule will be, that is 7.3 kilocalories of energy is stored in one ATP. So by the calculation of the amount of energies that are released by glycolysis and the citric acid cycle, and when it enters CTC, we have shown that it is 38 ATPs. Therefore, since 38 ATPs are generated and one ATP can conserve 7.3 kilocalories, the total amount of energy that we can get will be 38 into 7.3, which is 277.4 kilocalories. 277.4 kilocalories we will get because we get 38 ATPs are generated by one molecule of glucose. So what does thermodynamic efficiency therefore be for biological oxidation? If we do that reaction in the chemical reaction in the lab, we would have got 636 kilocalories, whereas we are getting only 277.4 kilocalories. So if I consider the efficiency, it just becomes 43%. 277.4 divided by 636 into 100, which becomes 43%, is only what is conserved by the combustion of one molecule of glucose into carbon dioxide water in biological systems. The remaining is waste. It is dissipated as heat. Even then, even when it is 43%, you know how much of energy we are gaining from glucose. If it was only one step, then we would have got only 7.3. Definitely, this is much more than having only one step reaction. Therefore, there are series of steps that are happening. So the final one is what we call it as the electron transport chain. As the name itself tells you, it is the transport of electron. So we call this as the final stage of aerobic respiration, which consists of series of redox reactions. So what are redox reactions? It is simply the electrons are passed on from a donor to acceptor molecules. When there has to be movement of this electron, we have to have what are known as electron carriers. And these electron carriers are called as cytochromes. Final link of this chain is the oxygen, which is the last acceptor of the electron. So the oxygen will accept the electron and the proton and form water. Hence, if oxygen is not present, the chain is cut. The oxygen has to be present to accept the electrons. And once the oxygen accepts the electrons and forms water, ATP will be produced by what is known as chemiosmosis. And this ATP is what is used as the energy. Hence, we should understand that this cannot happen in the absence of oxygen. So, the oxygen is life for all of us. Now, what happens in the electron transport chain can be organized into 4 large complexes. Please remember, whenever we are talking of any biochemical reaction, there will be the enzymes that are involved, which are nothing but proteins. So here we will have 4 large complex proteins, that is the enzymes, which are labeled as 1, 2, 3 and 4. That is 1 to 4. Now all the electrons that enter this transport chain, from where will they come? They will come from NADH. NADH is nicotinamide adenine dinucleotide, and FADH2, which is flavin adenine dinucleotide. So the nicotinamide and the flavin are supplied by the vitamins. So these are where they are produced. We already know they are produced in the cellular respiration. We already have shown where NADH and FADH2 are produced. They are produced in glycolysis, they are produced in citric acid cycle, they are produced in fatty acid metabolism. That is why I told you in the final stage, I'll tell you how these are converted into ATPs. So what happens is the energy that is released when these electrons transfer across these chains of the complexes 1 to 4, there will be an electrochemical gradient because the electron is moving. Remember, when electron is moving, there is electricity. So whenever there is mobile electrons, we'll always have a gradient. That is what we call it as the potential, the electrode potential. So whenever the electrons starts transferring from one part of the electron transport chain to another, as and when the electrons move, there will be protons that are generated. So it has to be, because totally it will be neutral. Hence, whenever there is a proton that is generated, this will be pumped into the inner membrane space. So there is an outer membrane of the mitochondria and the inner membrane of the mitochondria. In the inner membrane, the electron transport chain takes place and it pushes the protons into the space between the outer membrane and the inner membrane, where there will be a proton gradient. So as the electron moves in this membrane, the protons are pushed to the space and the protons start collecting in this space, and therefore there will be excess of proton or proton gradient. This proton gradient is important because once this proton gets collected and they are in certain concentration, then the proton will move into the ATP synthase or into the 4th complex. Water is formed and this proton gradient makes the ADP or the phosphate to get added to the ADP via the ATP synthase. I will explain with the transport chain now. I'll go to the next slide, which is the yellow. Okay, so now you can see this is our electron transport chain. I hope all of you can see this. Now you see this is our cytoplasm, and then we have the mitochondria, which has the outer mitochondrial membrane, and here we have the inner mitochondrial membrane. Of course, this is the matrix. Now in the inner mitochondrial membrane is the electron transport chain happening. That's why, remember, I told you that in the mitochondrial matrix is the citric acid cycle or the Krebs cycle. So all that which is given out from glycolysis or the metabolism of fatty acids or proteins will enter this matrix where the Krebs cycle takes place, and from the Krebs cycle, as soon as these are released, that is NADH and FADH2, will enter the ETC. So the ETC will be happening here in the inner mitochondrial membrane. We might have taken months together to discuss, but this is, remember, happening at every second. All right. So now what happens to this NADH that is released? Okay. Now that enters the electron transport chain through the complex 1. So this is, again, I told you, this is nothing but your enzyme, which is a protein. So when NADH gets delivered into the complex 1 and it gets converted to NAD plus, and the protons are released. These protons go into the inner membrane space. They are pumped into the inner membrane space where the electrons move in the mitochondrial membrane. Hope you are following. So there has to be electron carriers. So there will be the first electron carrier, which is called as the coenzyme Q, which is nothing but the ambiquinone. This is called as the ambiquinone, which will carry the electrons to the complex 3, not 2 but 3. I'll tell you why. Now, the FADH2, what we are talking of, the other electron carrier, which is also generated in the cellular respiration, will be pumped to complex 2. Why? Because the FADH2 is not as strong as NADH, so it is not possible for FADH2 to pump to 1. So there is another complex to which it will pump, and this complex 2 will send it to coenzyme Q, which is an electron carrier, and both of them will be pushed into complex 3. So now you see in the complex 1, when NADH pushes its electron and protons are generated, which is lost from FADH2. That is the reason NADH will give 3 ATPs, whereas FADH2 will give only 2 ATPs. Now, when NADH gives away its proton as well as the electron, it becomes NAD+, and NAD+ can again go into the Krebs cycle down into the matrix for again carrying out the reaction. Similarly here, FADH2 will push the protons and the electrons here and again get converted to FADH and get into the mitochondrial matrix. Now the electron starts moving into the complex 3, and once it goes to the complex 3, few more protons are pushed into the inner membrane space, and then the electrons move to the complex 4. Here the electron carriers are called as cytochrome C. So the cytochrome C will now push it to the complex 4. Here in the complex 1, 2, 3 and 4, what we are talking of, they all are proteins which will contain iron. So they have heme there, Fe2 plus to Fe3 plus. So what is happening, how they are carrying out electrons is, when I started, I showed you how Fa2 plus gets converted to Fe3 plus by releasing an electron. Like that, it can accept an electron and again become Fe2 plus. So this conversion of Fe2 plus to Fe3 plus, that is ferrous to ferric, by loss and gain of electrons, is what makes it carry the electrons. So that is how the electrons are carried. Once it comes to the complex 4, protons are again pushed into the inner membrane space and the oxygen. So 1 atom of oxygen, 2 protons and 2 electrons combine to form water. So in complex 4, 2 protons one oxygen atom, which is written here as half O2, and then 2 electrons combine, and you will get water. But remember, there will be all these protons that are in the inner membrane space. I am talking of one NADH. It is not one NADH that is pushed. Many NADH. Remember, 1 mole of glucose will be 6 into 10 power 23. So one molecule is giving so many NADH. 1 mole, even if it is reacting, so many NADH will come. So as and when they come here, they start pushing the electrons here, protons here. So what happens? There will be a proton gradient that will be developed here. Now, this proton gradient, which is present here, will generate ATPs by phosphorylating ADP by the help of what is called as ATP synthase. This complex 5 is nothing but ATP synthase. Now, what is this ATP synthase? This is something like a pump, like how the wheel runs. So, if you pour water, the wheel starts running, isn't it? So, because of the pressure of water, the wheel can run and generate energy. So, like how you get this hydropower, like that, the ATP synthase is acting like that wheel when the proton gradient will make it run and phosphorylate ADP to form ATP. So, because in 3 points the protons are generated, 1 molecule of NADH will generate 3 ATPs, whereas 1 molecule of FADH2 will release protons only in 2 complexes, and therefore only 2 ATPs are released. The release of ATP will depend upon this proton gradient. That is the reason we get 3 from NADH and 2 from FADH2. So, what is happening in the electron transport chain can be quickly revised if you go through this slide. So, first what is happening? Electrons enter the transport chain through the delivery by electron carriers NADH and FADH. These reduced electron carriers, which are obtained from the cellular respiration, will transfer the electrons in the beginning of the transport chain. So, the loss of electrons takes place. NAD plus and FADH is released, which is recycled into the respiration. Now, the NADH starts the process by depositing these electrons at complex 1, like I told you, and then they will release the protons into the space. Similarly, FADH2 will release its electrons and proton into the complex 2, and then it will go to the third complex. How the electrons that are coming from the complex 1 and complex 2 will then go to the ubiquinone, that is Q. That ubiquinone gets reduced to QH2, which is nothing but a mobile electron carrier. So it can become ubiquinone and ubiquinone H2. Therefore, it will transfer the electron into what? Into the complex 3. So as the electron pass through the complex 3, more hydrogen ions are pumped into the membrane, and then the electrons are passed through the cytochrome C, and then it will go to complex 4. Now in complex 4, it passes the electrons to the oxygen. So the oxygen is split into 2 oxygen atoms, accepts proton from the matrix and form water. So 1 oxygen atom, 2 protons and 2 electrons will form water. But the complexes 1, 3 and 4 use all the energy that is released when the electron is moving from higher to the lower energy levels, and to push the protons into the space, and therefore it will create the proton gradient. Now, once this proton gradient is formed, what happens next? That is the chemiosmosis and oxidative phosphorylation. So, the movement of these electrons through the electron transport chain, as I told you, will form the proton gradient because of the positively charged hydrogen ions, which will accumulate in one space, that is the space in between the 2 layers. Now, the hydrogen ions in this matrix will not pass through this. So only this hydrogen ions concentrated in the membrane will push into the membrane, that is into the inner mitochondrial membrane, and they will get embedded in the ATP synthase. So this ATP synthase, like I told you, will act like a water wheel. So it will allow the hydrogen ions to move down its electrochemical gradient. As it moves down, it catalyzes the addition of the phosphate into the ADP, generating ATP. This process is called as chemiosmosis. Energy from the proton gradient used to make ATP is called as chemiosmosis. And we see that in a eukaryotic cell, because of the cell respiration, 30 to 32 ATP molecules will be generated from one molecule of glucose. Only remaining 6 or so is generated in substrate level phosphorylation and otherwise. So most of the energy is generated by the electron transport chain and chemiosmosis. So how this is actually happening, you can see in this figure. The synthesis of the ATP from NADH and FADH2 through the electron transport chain and chemiosmosis is called as oxidative phosphorylation. So you can again see here how we have the complex 1, then complex 3 and complex 4. So the complex 2 is somewhere here, only which accepts the electrons from FADH2. So NADH will pump in one, forming NAD plus. So you can see this is our membrane in which the electron transport chain is happening. This is the inner membrane space where the protons are thrown. This is our matrix where the other processes are taking place. So then it carries, this is our first carrier, that is ubiquinone, and then it pushes to complex 3. In all these complexes we will have Fe2 plus to Fe3 plus, Fe2 plus to Fe3 plus is what is happening here. And then again the electron carriers are here, these are cytochromes which will carry electrons to 4. So you can see the proton gradient here. Now here the water is formed, and because of this proton gradient, which will now push the protons into this is our ATP synthetase, which will synthesize the ATP. So this part is electron transport chain. This is our chemiosmosis. So together, the electron transport chain and chemiosmosis have generated ATP from NADH and FADH2, and this is called as oxidative phosphorylation.